Hi, everyone. Welcome to Naturally Man for Life. Thank you for joining me today, and thank you so much for your time. Today, I have what I'm going to just call a fluff piece, which is about the microbiota, your second genome. I thought you might be interested to learn just how much these types of substances affect components and substances affect your overall bodily functions. And this is primarily regarding autoimmune conditions, mostly RA, lupus, and assorted other arthritic type conditions like ankylosing spondylitis, psoriatic arthritis, and a few others. So let's get started. But first, an anecdotal piece you might like to hear. I did have a dentist appointment. This is regarding the xylitol. I did have a dentist appointment last week, and I'm happy to say I was in and out of there in 20 minutes. So that's just strictly anecdotal, but um, it's just, it made me happy. So I'm just going to continue doing what I do and using what I shared with you in the last posts. So, the gut microbiota, your second genome. Now, I did not include very much about this new let me see what they're calling it. Some type of carbon. Um, an aryl hydrocarbon receptor, which is offering promising insight into these conditions. So, okay, that's the only really new thing. But this is from 2019, so that's what they're focusing on. So, the gut microbiota, your second genome. All mammals, including humans, are born from a sterile environment inside the womb. Right after birth, microorganisms begin to colonize different parts of the body, such as the skin, mouth, nose, and especially the digestive tract. These microbes form communities that live on surfaces of tissues covered by epithelial cells. One of the most important of these communities is the gut microbiota, a large population of microbes that live in the intestines and work in a close cooperative relationship with the human body. What is the gut microbiota? Gut microbiota refers to all microorganisms living in the digestive tract. This includes bacteria, viruses, fungi, protozoa, and archaea. While all of these play a role, bacteria are the most widely studied. These gut bacteria help the body in many ways and are a key part of human health. Some bacteria are beneficial, some are neutral, and others can become harmful under certain conditions. The gut is the body's largest point of contact with the outside world through food and water, and it holds about 2/3 of all microbes living in or on the human body. In total, the human body carries more than 100 trillion microorganisms with the majority residing in the intestines. The development of the gut microbiota begins at birth and continues into early childhood. This early development shapes the immune system and long-term health. If the natural process of gut colonization is disrupted by things like unnecessary antibiotics, poor nutrition, or lack of breastfeeding, including early weaning, it can lead to long-term health problems. There are three main stages in the development of the infant microbiota. The developmental phase is from 3 to 14 months. The transitional phase is from 15 to 30 months, and the stable phase is from 31 to 46 months. So that's almost 4 years of age. Breastfeeding plays the most important role in shaping a healthy gut microbiome during these early stages. Human milk contains not only nutrients, but also prebiotics and antibodies that help build a strong microbial foundation. Functions of the gut microbiota include digestion and metabolism. The microbiota helps break down complex carbohydrates, synthesize vitamins, and absorb nutrients from food, supporting energy metabolism. Detoxification. Gut microbes help neutralize harmful compounds that enter the body. Immune system development and support. A balanced gut microbiota trains the immune system to distinguish between harmful and harmless substances. Inflammation control. Healthy gut bacteria help regulate inflammation, which is essential for preventing chronic diseases. Gut microbiota can be grouped in how they interact with oxygen. Most of the microbes in the gut are obligate anaerobes, meaning they thrive in oxygen-free environments like the colon. There are also facultative anaerobes, which can live with or without oxygen, and aerobic bacteria, which require oxygen to survive. Microbial diversity in the gut. The most common bacterial groups found in the human gut are firmicutes, bacteroidetes, actinobacteria, and proteobacteria. Thanks to new DNA sequencing technologies, researchers have discovered that the gut microbiome contains about 3.3 million genes, around 100 times more than the human genome. Because of this, scientists often refer to the gut microbiome as the second genome. It's also referred to as the second brain. Functional roles of gut microbes. The gut microbiota is made up of a variety of microorganisms that can be grouped based on their function and impact on health. Some microbes are considered commensal or beneficial, meaning they support the body by aiding in digestion, producing vitamins, and strengthening the immune system. These bacteria and regulating the immune system. These bacteria live in harmony with the host and help maintain a stable internal environment. Another group includes opportunistic organisms, which are usually harmless, but can become problematic when the balance of the microbiota is disturbed, such as during illness or after antibiotic use. There are also pathogenic microbes, which are harmful and capable of causing disease when they become dominant. In contrast, therapeutic microbes, often introduced through probiotics, can help restore balance in the gut after it has been disrupted. Together, these groups interact in complex ways that influence overall health disease resistance, and inflammation control. The composition of the gut microbiota is not the same for everyone and can change significantly over a person's lifetime. A number of factors influence which microbes are present and how they function. Diet is one of the most powerful influences. For example, diets high in fiber tend to promote the growth of beneficial bacteria, while highly processed or high-fat diets can encourage harmful microbes. Age also plays a role, with microbial diversity generally increasing from infancy to adulthood and often declining in older age. Sex may affect microbiota composition due to hormonal differences, and geographical location can shape the microbiota based on local diets, cultural practices, and environmental exposure. Additionally, medications, especially antibiotics, can drastically reduce microbial diversity, often wiping out both good and bad bacteria. Even non-antibiotic drugs have been shown to affect gut microbes. Health conditions such as obesity, diabetes, and gastrointestinal disorders can further disrupt the microbiota, leading to long-term imbalances. Finally, the use of probiotics, prebiotics, and other microbial therapies can help maintain or restore a healthy gut flora when used appropriately. Maintaining a healthy gut microbiota is essential for overall health. Disruptions in this ecosystem, whether due to poor diet, stress, illness, or medication, can lead to a wide range of health problems. In recent years, an increasing number of studies have shown that several approaches, including antibiotics, prebiotics, antimicrobial treatments, fecal microbiota transplantation, and specific probiotics, can help regulate the gut microbiota. However, inappropriate use of antibiotics and even some non-antibiotic drugs designed for human use have been linked to negative changes in the gut's microbial composition. There is growing experimental and clinical evidence that long-term imbalance or dysbiosis in the gut microbiota can trigger an inflammatory response. This inflammation has been strongly associated with the development of autoimmune diseases, especially in people who are genetically predisposed. Interestingly, germ-free animal models, which are raised without any exposure to microorganisms, have become important tools for studying how the microbiome influences the development of many disorders. These models help researchers understand how gut microbes can either trigger autoimmunity in some individuals or protect against it in others. Overall, protecting and nurturing the gut microbiota through thoughtful lifestyle choices, proper medical care, and possibly targeted therapies may play a powerful role in maintaining long-term health and preventing disease. So the gut microbiota and autoimmune diseases. Research has highlighted the gut microbiota as a key player in the development of autoimmune diseases. Autoimmune conditions marked by the abnormal production of autoantibodies and inflammatory immune cells can be triggered by both genetic and environmental factors, notably shifts in the gut microbiota, often caused by modern lifestyle and dietary habits, and the widespread use of antibiotics may contribute to the rising incidence of these diseases. The gut's commensal microbes form a complex ecosystem that strongly influences immune function. When this balance is disturbed or dysbiosis occurs, that can drive autoimmune responses. While the exact mechanisms remain unclear, pathways involving immune regulators like aryl hydrocarbon receptor offers promising insight. As a result, therapies that target and restore gut microbiota balance are emerging as a potential strategy for preventing or managing autoimmune diseases. So, specifically, how this works by acting on aberrant production of autoantibodies. They cause abnormal generation of autoantibody producing B cells and autoreactive T cells, and anomalous production of pro-inflammatory cytokines. It has been hypothesized that the increasing incidence of autoimmune diseases is due to considerable shifts in the microbiota among multifactorial reasons following dietary changes and the widespread application of antibiotics. Tolerance also plays a key role in collagen-induced arthritis in rats. The intestinal mucosal immune response is enhanced and immune tolerance is disturbed. Moreover, madecassoside treatment, which I don't know what that is, can increase relative expression of forkhead FOXP3 microRNA in the small intestine, downregulate concentrations of secretory IgA and interferon in the small intestine content and tissue respectively, and decrease the ratio of CD4 and CD8 cells in the epithelium and lamina propria, and decrease relative expression of CD80, CD86, IL6, and IL12, and microRNA collagen-induced arthritis rats to downregulate the intestinal immune response and restore normal immune tolerance. So there are some things they know how they work and some things they don't. So milk, what you can do, milk is a postnatal imprinting system, stabilizing FOXP3 expression and regulatory T cell differentiation. Accumulating evidence underlines that milk is a complex signaling and epigenetic imprinting network that promotes stable FOXP3 expression and long-lasting tract differentiation, crucial postnatal events preventing atopic and autoimmune diseases. So, okay, I don't know why it's getting darker in here because it's getting later and it's cloudy out and I didn't turn the light on. So sorry about that, but okay, so that's the end. And if you want the source, it's included. But it was extremely, extremely long, and it was interesting. One interesting note is that in lupus and rheumatoid arthritis, it highlighted the effects of smoking on severity and damage to the joints and organs and bones caused by the autoimmune triggering response. Okay, everyone, I thank you so much for joining me today, and I will see you next time. You have a great day and a great week. Bye.